Catabolism of amino acids. The catabolism of amino acids involves 3 stages. The first stage, in which there is going to be removal of the amino group from the amino acid. Second stage involves disposal of ammonia in the form of urea through urea cycle. And the third stage involves the catabolism of the carbon skeleton of amino acids. The first stage, in which the amino group is removed from the amino acid, can be with the reactions that involves the transfer of the amino group, which is called as transamination reaction, or it can be with the deamination reaction. Now this deamination reaction can be an oxidative reaction or a non-oxidative reaction. We will see about all these stages in detail. First we will see about the transamination reactions. Transamination reactions are going to be catalyzed by the enzyme transaminases, which are also called as aminotransferases. These aminotransferases or transaminases are going to help in the transfer of the amino group from an alpha amino acid to an alpha keto acid. Now this reaction will result in the formation of a new alpha amino acid and a new keto acid. Let us take the example of an alpha amino acid reacting with alpha ketoglutarate. Now the amino group from the alpha amino acid will be transferred to the ketoglutarate, and the keto group from the alpha ketoglutarate will be transferred to the alpha amino acid, which will result in the formation of the L-glutamate and a new alpha keto acid. Some of the most common transaminase enzymes are the alanine transaminase and aspartate transaminase. Alanine transaminase helps in the transfer of the amino group from the alanine to the alpha ketoglutarate, resulting in the formation of L-glutamate and pyruvate. Since this enzyme helps in the formation of pyruvate, it is also going to be called as glutamate pyruvate transaminase, GPT. The other important transaminase is the aspartate transaminase. Aspartate transaminase helps in the transfer of amino group from the aspartate to the alpha ketoglutarate, resulting in the formation of glutamate and oxaloacetate. Now this enzyme is also called as glutamate oxaloacetate transferase, in short GOT, not Game of Thrones, but glutamate oxaloacetate transferase. Now in this transamination reaction, there is going to be no net loss of any of the amino groups. They are only going to be transferred from the amino acid to the alpha keto acid, and then there is going to be resulting new amino acid and new alpha keto acid. Now they are all going to be forming one common product, that is L-glutamate. All the amino acids except lysine, threonine, proline and hydroxyproline participate in the transamination reactions. Significance of transamination reaction. The transamination reaction provides a mechanism through which the amino groups of various amino acids are going to be condensed into one amino acid, that is the L-glutamate. Now L-glutamate can act as a amino group donor for the synthesis of non-essential amino acids in our body. The aspartate transaminase and alanine transaminase act as markers for the impairment of liver function and also as markers for cardiac function. Deamination. The process of deamination can happen through two different processes. One is an oxidative process and another one is the non-oxidative process. The oxidative process of deamination can take place with the help of the enzyme glutamate dehydrogenase or by the help of the enzyme amino acid oxidase. The non-oxidative deamination can take place with the help of amino acid dehydratase enzyme. Oxidative deamination by glutamate dehydrogenase enzyme. Glutamate dehydrogenase enzyme helps both in the catabolism of amino acids as well as in the anabolism of amino acids. In the catabolism of amino acids, glutamate dehydrogenase helps in the deamination reaction, where the amino group is removed from the L-glutamate to form alpha ketoglutarate and the amino group. Now this reaction is going to be helped by the reducing equivalent that is NAD plus, which is going to be converted to NADH. Whereas in the anabolic reaction, it is going to be in the opposite direction. Alpha ketoglutarate will combine with an amino group to form L-glutamate in the presence of glutamate dehydrogenase enzyme, but helped by the reducing equivalent NADPH, which will be converted to NADP plus. So glutamate dehydrogenase has both the anabolic role as well as the catabolic role. Now this is the first oxidation, that is the oxidative deamination by glutamate dehydrogenase enzyme. Glutamate dehydrogenase enzyme is also elevated in cases of the impaired liver function. Oxidative deamination by amino acid oxidase enzyme. Amino acid oxidase enzyme requires the flavin coenzymes such as FMN and FAD. Now this amino acid oxidase is going to help in the deamination of the alpha amino acid. So alpha amino acid is going to be first converted to an amino acid, and then the amino acid is going to help in the formation of an alpha keto acid. In this process, there is going to be removal of the amino group from the alpha amino acid. In the conversion of the alpha amino acid to alpha amino acid, there is going to be the reducing equivalent FMN or FAD, which are going to be reduced, and in this reduction reaction, there is going to be a coupled reaction which is going to generate the hydrogen peroxide. Hydrogen peroxide, which is generated in this reaction, can be neutralized by the enzyme catalase. Non-oxidative deamination of amino acids with the help of amino acid dehydratase enzymes. The alcohol containing amino acids such as serine and threonine, because of the presence of the hydroxyl group in them, can be directly deaminated to form ammonia. Serine with the help of serine dehydratase enzyme can be split into pyruvate and ammonia in the presence of pyridoxal phosphate. Threonine with the help of threonine dehydratase enzyme can be split into alpha ketobutyrate and ammonia in the presence of the pyridoxal phosphate as the coenzyme. The metabolic fate of ammonia. Ammonia that is formed by the end of transamination and deamination reactions are highly toxic in nature. Therefore, this ammonia is immediately converted to non-toxic metabolites before they can be transported to liver or kidney. They are transported in two forms. One is the glutamine and then the other is the alanine. Glutamate can combine with ammonia in the presence of glutamine synthetase, which is an energy requiring process. Therefore, ATP is also required for this conversion. So glutamate combines with ammonia in the presence of glutamine synthetase to form glutamine, and this glutamine that is formed is going to be a non-toxic metabolite. Now this glutamine can cross the cell membranes easily, whereas glutamate cannot cross the cell membranes. Glutamine can be transported to the liver, and in liver glutamine can be again converted back to glutamate and ammonia with the help of the enzyme glutaminase. Glutamine is the major form of transportation for the ammonia in our body.